Greetings everyone. Today in this session we are going to discuss about the antibody production theories. There are some theories which discuss about the how antibodies are produced in our body. That is, specifically we are going to discuss about the antibody production in the human body. So before going to theories, there are some basic concepts that we are going to discuss. First of all, what are the antibodies? Antibodies are the Y-shaped proteins present into our immune system. These Y-shaped proteins are produced through the immune response. So basically, upon encounter with the antigen, what are the antigens? Antigens basically are the foreign particles to the host body against which the body generates the immune response. So the antibody production takes place through the humoral immune response. In previous lecture, we have discussed about the immune response generated through the human body. That is, one is a cell-mediated immune response or cellular immune response, and another one is humoral immune response. The cell-mediated immune response includes the two types of cells. One is CD8 plus cells, CD cluster of differentiation. The CD8 plus cells are also known as T cytotoxic cells. Then there are CD4 plus cells, that are the T helper cells, which helps the other lymphocyte for the generation of immune response. Then there is humoral immune response. In humoral immune response, there is production of antibodies through the B lymphocytes. All regarding the generation of humoral and cellular immune response is already discussed in previous lecture. So today we are only focused on to the humoral immune response. In humoral immune response, the B plasma cell generates the monoclonal antibodies which are specific against that specific kind of antigen which is encountered into the host body. So before going to discuss the theories of antibody production, there are some basic concepts regarding the antigen and antibody. Antigen can be a bacteria, can be a virus particle, can be a fungi, can be a protein, can be a small dust molecule in case of hypersensitivity. There are vast range of antigens against which our body can generate the immune response. So suppose this is an antigen, suppose this is a virus particle. The virus particle has the glycoproteins on their surface, and these glycoproteins are responsible for the generation or the establishment of infection or disease into the host. What the antibodies does, because we have already discussed that the antibody works by three means. First is neutralization, second one is opsonization, and third one is complement protein recruitment. So either by opsonization or either by complement recruitment or either by neutralization, the antibody blocks the antigen, or antibody cancels the action of antigen, due to which the establishment of infection or the disease is prevented. So this is virus particle, suppose this is glycoprotein. The antibody blocks the active site of that antigen, and then the infection can be prevented. So question is that how antibody identifies that antigen, because the antigen and antibody shows the complementarity reaction against each other. Then and then only when the complementary fit is established in between antigen and antibody, these complexes are formed, and the antigen antibody complex is responsible for the generation of immune response, so through which the antigen is eliminated in our body. So this is basic concept about the antigen and antibody. So how these antibodies are made, how these antibodies are produced. So there are some theories regarding that. So theories basically are of two types. One theory is instructive theory and another theory is a selective theory. So instructive theories suggest that antigen itself directs the immunocompetent cells. Immunocompetent cells are the cells which are able to generate the immune response, specifically humoral immune response. So antigen directs the immunocompetent cells to generate the specific kinds of antibody against itself, while the selective theory suggests that there is no role of antigen into the generation of specific kinds of antibody against that antigen, but the antibodies are already produced inside the host prior encounter with the antigen. So Again, in instructive theories, there are two kinds of theories, direct template theory and indirect template theory. While in case of selective theories, there are three theories, side chain theory, natural selection theory and clonal selection theory. We are going to discuss one by one. First of all, direct template theory, which is included into the instructive theory. Brinell and Horowitz suggested that upon encounter of the antigen, the antibody fold itself against that antigen. So basically, antibodies are the protein molecule. How proteins are generated? Through the generation of peptide chain. These peptide chain are then folded into the specific kind of protein, that is the Y-shaped protein. First point, we have discussed that proteins are the antibodies are the Y-shaped proteins. So direct template theory suggests that upon encounter of the antigen, the protein molecule, that is a peptide molecule which is going to be formed into an antibody in future, folds itself against that antigen, and then only the antibody specific to that antigen is generated. But this theory has also a flaw. Take an example of antigen and antibody. Suppose there is an X antigen. The antibody produced against that X antigen, suppose it is IgG, immunoglobulin G. So according to this theory, which suggests the antibody itself fold against that antigen. So upon encounter of X antigen, IgG folds itself. The protein can be unfold or refold into the cellular level due to any kind of reason. So for any reason, suppose this antibody is unfolded and refolded. So upon a refolding, it is first unfolded and then refolded. Upon refolding, if we introduce a new kind of antigen, suppose that is Y antigen, so what will happen? Here, upon refolding, the antibody regains its original shape, that is against that X antigen. So how this theory explains this phenomena? Because at the time of refolding, we are going to introduce a new kind of antigen. But upon refolding, the antibody regains its original structure, regains its original specificity, which is generated against the X antigen. So this kind of theory is also discarded. Next one is indirect template theory. The indirect template theory is given by Burnett and Fenner in 1949. This indirect template theory suggests that antigen first enter into the B cell, B lymphocyte, and then the genome of the antigen is incorporated into the immunocompetent, that is B cell's genome, and then this genome is transcribed, translated, and the protein formed from this translation process is then folded into that antibody, and this antibody is specific to that antigen. And these cells now proliferate itself or stimulate the cell to generate the numerous amount of these types of antibodies. Again, this theory is discarded because it has flaw, because this theory does not explain about the secondary immune response. There is primary immune response in which the first antigen is encountered into the host cell, it is eliminated. In secondary immune response, the similar type of antigen is encountered, and in case of secondary immune response, the response generated through the host body is more rapid. So this theory does not explain the secondary immune response, how the secondary immune response is generated and it is regulated. So these two are the instructive theories which suggest that antigen itself instructs the immunocompetent cells to produce the antibodies, and both are the wrong. Next, we are going to discuss about the selective theories. So in selective theory, first one theory is side chain theory. The side chain theory is given by Ehrlich in 1898. According to side chain theory, the immunocompetent cell, that is B lymphocyte, bears diverse range of side chains. Side chains, now we know these side chains are the BCR. Now we know these side chains are BCRs. What are the BCRs? BCRs are the B cell receptors. So, according to side chain theory, B lymphocyte have a diverse range of side chains onto their surface. So, these side chains can combine with or can show complementarity with the encountering antigen. So the antigen can come in future in the host body, and then the antigen which shows antigen combines with the antibody which shows the perfect fit against it, and then only the activated B cell. Here the activation of B cell takes place. We have already learned about the activation, proliferation and production of the B cell. So when the antigen encounters to the B cell, it gets activated, and upon activation of the B cell stimulates itself to produce the same kind of side chain which shows the perfect fit against that antigen. So this is all about the side chain theory, but again there is flaw in this theory, because this theory does not explain about the immunity to the synthetic antigen or the artificial antigen, which can be nanoplastic or new chemicals which are developed or generated through the humankind. So this side chain theory is also discarded. Next, the next theory is natural selection theory, given by Jern in 1955. The Jern suggests that the antibodies are formed in the embryonic life. In the embryonic life, million copies of antibodies are already formed and they circulate in our bloodstream. And upon encounter with the antigen, the antibody which shows the specific complementarity against that antigen get interact with each other, antigen antibody complex is formed, and that antigen is eliminated through our body. But there is also flaw in this theory. First of all, this theory suggests that there is generation of a pool of antibody in our bloodstream, but there is no such generation of antibody pool in our bloodstream, because the antibody are only and only released in our bloodstream after the generation of immune response. So what is the process? Upon encounter of the antigen first activates our lymphocytes, B lymphocyte, T lymphocyte, then B lymphocyte is proliferated, is expanded into the B plasma cell, and then only B plasma cell generates and secretes the monoclonal antibodies into our bloodstream. So this theory, natural selection theory, is also discarded, but the scientist Jern contributed into the immunology in very much higher level. The Jern gave total 3 theories. First one is natural selection theory. Then there are 2 more theories which specifically discuss on the immune network theory, and he got Nobel Prize for that in 1984. So this is all about the natural selection theory. The last one, and which is widely accepted through all over the world, that is how antibodies are produced. That theory is called as clonal selection theory, and the theory is given by Burnett in 1959. The clonal selection theory has multiple steps which explains how antibodies are made in our body to eliminate that pathogen and how the humoral immune response is generated. So first of all, clonal selection theory suggests that there is a generation of different kinds of or different types of clones of the B lymphocyte. These are the clones of the B lymphocyte. So how these clones are formed, how the different types of clones are formed, that is another topic that will be discussed in another session. So according to clonal selection theory, different types of B cell clones are produced through the hematopoietic stem cells. Hematopoietic stem cells which generates the lymphoid progenitor, which generates the B cell progenitor, and finally which generates the B cell. Each B cell generated through the hematopoietic stem cells or the progenitor have a different binding site or have the different active site on their BCR. What are the BCR? BCR again the B cell receptor. It is nothing but the antibodies. So antibodies, before encounter of the antigen or before generation of the immune response, the antibodies are only present onto the B cell that are known as the BCR. So each and every BCR has a unique active site, has a unique antigen binding sites. So these are called as clones. Next, among that clone, whenever a antigen enters into the human body, the specific type of BCR which shows the complementarity fit against that antigen is selected. The BCR is selected means that B cell is selected for the generation of immune response. Suppose this cell is chosen against that antigen because it shows the complementarity fit against it. Then this process is called as clonal selection. This process is called as clonal selection. After clonal selection, a single B cell is chosen for the production of single type of antibody, and these antibodies has the ability to eliminate that antigen which is priorly encountered into the host body. Then the next process is clonal expansion. In the clonal expansion, the selected B cell or the activated B cell rapidly multiply itself. So there is a rapid generation of the progenies of the selected B cell clone. So different numbers of single type of B cells are generated. Single type of clones are generated. In the process of clonal expansion, the cells are differentiated into two categories. First category is the generation of effector cells. Effector cells are the B plasma cells. These cells are the effector cells or the B plasma cells. And the rest cells are included into the memory cells. Specifically, B plasma cells are larger in size and which has the multiple units of endoplasmic reticulum, because these B plasma cells are programmed to produce the numerous amounts of antibody through their cells. And some cells store the information of that antigen, and these cells are known as the memory cells. So in this way, humoral immune response is generated, and this theory explains both primary immune response and secondary immune response. So primary immune response is basically generated through the B plasma cell, while the secondary immune response is generated through the B memory cells, through the differentiation process. And this is all about the clonal selection theory. Here one question arises that how these clones are made, how the different types of BCRs are made. There are multiple concepts how the different types of BCR, how the different kinds of active binding sites of that antibodies are generated, which will be discussed in another session. So this is all about the antibody production theories, the instructive theories, the selective theories. In instructive theory, there is a direct template theory, which suggests that antigen folds the antibody. Indirect template theory, which suggests that the genome of the antigen is incorporated into the genome of the immunocompetent cell, and then antibodies are produced. Then there are selective theories. In selective theory, there is side chain theory, which suggests that the B cell has the multiple side chains present onto their B cell. Before the encounter of any antigen, already the different kinds of side chains are already present into the cell. Then a new antigen came, and the side chain which shows the complementarity fit against it, then it forms the antigen antibody complex, and the cell stimulates itself to produce the same kind of side chain, that is the same kind of antibody through that cell. Then there is natural selection theory. Natural selection theory suggests that in the embryonic life of the human, already million trillion copies of the antibodies are already produced, which circulates in our bloodstream, but which is also not true. And lastly, there is the clonal selection theory. Clonal selection theory which says there are some clones. The clones are selected, they get expanded through the rapid cell division, and then there are differentiation of that clones, of that expanding B cell, which explains the generation of primary immune response and secondary immune response. Thank you.